Okay, so the next filament I want to talk about is microtubules. Now these are the largest in diameter of all three cytoskeleton components that we're going to be talking about. So let's look at their structure. First of all, they're made up of a bunch of little heterodimers, so two different little subunits, the alpha and beta. So I put the beta in red over here and the alpha in blue, and then you add them all together and you create the string that we call a protofilament. Now, an important thing about microtubules is that they have a polarity, which means they have one side that is the minus end and one side that is the plus end. So the minus end would be the alpha, and then the plus end is the beta. So the minus end is going to be like stable. It's anchored in the organizing center, which we'll talk about in a second. And then after that, the plus end in red is where we're going to be adding on these heterodimers. So another important thing about microtubules is that they're dynamic. So they can grow and shrink depending on what you need them to do. So when you're adding or taking away these dimers, you're going to be doing that on the plus end. So all these protofilaments join together and they form this hollow tube that we call tubulin. Now let's look at where this is used in the cell. So in polarized cells, which means cells that don't have like a perfect shape, you kind of stretch them out a bit, you have these capping proteins on the side that you want to eventually become like the apical side, say if we're talking in epithelial columnar cells or columnar cells. Those are kind of column shaped. So you want them to have that apical side. So you would have capping proteins on that side, and then microtubules, dynamic, so they're going to grow from the plus side until they reach those capping proteins, and then they'll be anchored there, and they will have stretched the cell kind of so that it gives it that shape. And then there's some there is some microtubules at work during mitosis as well, because they actually form that those spindle fibers. They're actually microtubules that will go and grow by adding those dimers onto them, and then hold onto those chromosomes, split them apart, and then divide them into two different cells. So when you're pulling them back, again, you're taking away those dimers, always adding and taking away from the plus side of the tubulin. So these are temporary because you're not always doing mitosis, right? But then a more permanent form of microtubules would be found in epithelial cells, or really any cell that has cilia or flagella. So I drew cilia over here. And so these are the more permanent type, and they're actually giving the structure to that ciliated part. So microtubules are anchored in an organizing center, which is pretty much where they start off. That minus center has to be somewhere. So when you're talking about cilia or flagella, they're going to be anchored in a basal body, forming kind of like this basement thing, so that the microtubules can go above and create the cilia. And then here it would be the centrioles. So we talked about how microtubules are important for giving cell shape, for giving cilia and flagella, as well as during mitosis. They also play an important role in anchoring organelles into their plate. Now we're going to talk about how they interact with motor proteins. So let's look at a neuron over here. So along the axon, say we have proteins made over here, and we want to send them out to be excreted into the postsynaptic cleft to be sent to another neuron. So we're going to get them from the cell, and they're going to be in a vesicle. Now that vesicle, to be able to get to the other side, remember this over here in pink is a microtubule. So this is the minus end. The minus end is always where the cell is. And then after that, the plus end is the edge, like leaving the cell. So once you're moving towards the plus end, there's a specific motor protein that you need that only moves in that direction, and that is kinesin. And the other one that goes in the opposite direction is dynein. Now this one goes towards the minus end, but they are both carrying whatever cargo they need to carry. Both have globular heads. Now, these heads have ATPase activity on them, which means they're enzymes that can use ATP because when you're moving these motor proteins along this microtubule, you need ATP to generate the energy for that movement. So their heads have those ATPases. And then after that, these tails are the two top parts, and that's where you would be attaching the cargo onto. So dynein is also much faster than kinesin, and they move. There's what's responsible for the movement in flagella and cilia.